Okay, so now I have added those bases in just like I was doing before, and again remember that it just keeps adding it all the way up until it gets to that point where helicase has to split it open. And again, just like we did before, we need to look at that other side. It's just really important that you understand that once that primer gets in there and the new DNA polymerase starts adding the bases on, it can just keep adding and adding and adding towards the split, towards where the DNA is being unzipped. It doesn't have to stop because it can only add to that 3' end, and there's always going to be that 3' end there. Let's look at the other side here real quick. I'm just going to fold this one leg out of the way. Remember, we had to start here and work our way this way because DNA polymerase can only add to that 3' end. So this side has to wait until there's enough bases that have been unwound, enough bases that have been unzipped to where it can come back in and add nucleotides way up here so that it can then turn around and add them back in the correct direction so it has a 3' end. So DNA polymerase will have to jump, so it's going to have to stop doing the replication. It's going to stop doing the replication from this end and then have to jump up to a part where it can start that replication process. So on this side of the DNA strand, it's going to stop and start and stop and start. Where on the other side, once it gets started, it can keep on going. It doesn't have to wait. Now the complement to thymine again is going to be adenine. DNA polymerase can add in here. It's not an issue. And the reason why is because we've got a 3' end, we've got a base here that it can add to, a complement it can add to, so it's just going to add it in. And remember, to hook these things together, to hook this new strand that's being formed together, it has to be added together with the phosphate and the number 3 carbon on that sugar. This is why DNA polymerase only adds to the 3' end. It's the only way it can get these pieces to go together. And again, we're going to form our bond here. And DNA polymerase is going to move away from that origins of replication. It's going to move away from that spot where the DNA has been unzipped. And it's important to note that this side, it does move away from that spot where it unzips. Let me grab another guanine nucleotide in here being added because we have that open 3' end available to make that bond so that we can put this strand together in a nice big long sequence. So I'm going to go ahead and add the rest of these on to the to this strand. Okay, and we'll be right back.